Here. Good afternoon, everyone. I'm Gaetan, and I'm with Ever Hernandez Olivas and Elise Charton. Elise, you can turn on your camera. Hi. We are delighted to welcome you to our 6th Infogest Viva Webinar. This event was organized by a team of postdocs in the Food Research Center of Moorpark, Teagasc, every first Wednesday of each month. The goal of this webinar is to provide you the most recent findings in the field of digestion from the PhD Viva defended over the last year, and to maintain, of course, the link with the Infogest community. This webinar is broadcasted live on YouTube and will be accessible on the Infogest YouTube channel 2-3 days after the webinar. Elise Charton, I can pronounce it French way, is our speaker today. Elise successfully defended her PhD in February in the Agronomic Institute STLO of Rennes Angers in France. Her project focused, among other things, on whether mother's milk is digested differently to infant formula, on the role of tryptophan and how it's monitored, and on the effect of the gut-brain axis. She is now on a 3-month postdoctoral position to finish the writing her PhD articles, and she prepared a pre-recorded presentation. The presentation will be followed by a live question-answer session, so I'm inviting you to use the Q&A button at the bottom of your screen, and we will now start the presentation.
Human milk versus infant formula, protein digestibility, and impact on the microbiota-gut-brain axis. This work was supervised by the STLO and l'Institut Numécan in France, but also co-supervised by the Redet Institute in New Zealand. In this study, we were focused on human milk, which is frequently defined as the gold standard of infant nutrition in literature, and because despite of the exclusive breastfeeding recommendations up to 6 months in the world and up to 4 months in France, only 48% of infants under 6 months of life are exclusively breastfed, with discrepancies existing between countries, and that whereas the breastfeeding rate increased in France during the last decades, only 14% of infants were exclusively breastfed at 4 months of age, and this rate decreased to 1% at 6 months of life. The differences between countries and the low exclusively breastfed rate in France can be due to different reasons. The first one is that the mother don't want or cannot breastfeed their own child, but also can be explained by different cultural and socio-economical reasons, and also can be explained by the access to infant formula. For example, in France, the infant formula market is highly developed. Indeed, France is one of the first European producer and exporter and allow the supply of high-quality infant formula products. If we are looking at the composition of human milk and infant formula, it has been highlighted in that literature that they had different fine composition and structure. Indeed, human milk is a complex bioactive fluid which composition varies across time, in contrast to infant formula, which composition is defined and highly regulated in Europe. Infant formulas are mostly based on cow milk, which requires the addition of whey proteins to rebalance the whey protein to casein ratio and better mimic that of human milk. If we are looking at the overall macronutrient composition of human milk according to an average of literature data in comparison to the European regulation composition of infant formula, we found that we had a similar macronutrient composition, but that, for example, for fat content, the fine composition differs, notably due to the different milk origins, and also for the gray part, that this fraction is more complex in human milk with the presence of some bacteria or non-nutrients such as growth hormone, for example, that are missing in the European regulation. Moreover, the crude protein content was in a similar range in human milk and infant formula, but its fine composition differs. Indeed, the crude protein content is composed of the true protein and the non-protein nitrogen contents, for which the true protein content is higher in infant formula than in human milk, with difference observed in fine protein compositions, but also because the non-protein nitrogen fraction is higher in human milk than in infant formula, with also differing fine composition of this non-protein nitrogen fraction. Today, there is a higher true protein content in infant formula than in human milk. Infant formula are made to replace the human milk, and that's why the objective is to mimic as closely as possible the human milk aminogram in terms of amino acid profile, but also in terms of digestibility. But today they only made on the hypothesis that amino acids are 100% bioavailable. The notion of bioavailability represents the nutrient fraction digested, absorbed by intestinal mucosa, and available for subsequent organism metabolic functions. But if we are looking at in literature, only a few studies were focused on the digestibility of amino acids, and they highlighted that depending on the amino acid assay and study, the digestibility differs, with missing data for tryptophan in the in vivo study due to the difficulties of tryptophan determinations, which method differs from the other amino acids. And these two informations, the amino acid profile and the digestibility, are both combined to determine and evaluate the protein nutritional qualities through the Diaz determination, such as previously determined in the FAO report. Further investigation is needed regarding the determination of amino acid digestibility, with actually only one study in vivo with no data for tryptophan. So we were focused on tryptophan in that study because it's an essential amino acid that acts in the microbiota-gut-brain axis. And the tryptophan is mainly used in the protein synthesis, but can be also metabolized to the 5 other persons in different host pathways. So the first one is the Comunian pathway, which will lead to the production of some NAD+ and further niacin, or acetyl-CoA, that are both involved in some energy metabolism. But the tryptophan can also be metabolized by the host in the serotonin pathway, which contribute to the gut motility in the whole body and at brain level, depending on the tryptophan enlarged neutral amino acid competition, can be metabolized into serotonin and further in melatonin that are involved in food intake, mood, and sleep regulation. Furthermore, tryptophan can also be metabolized through a bacterial pathway, and especially called the indole pathway. In addition to difference in composition, infant diets differently modulate the health and the microbiota-gut-brain axis. Indeed, breastfeeding was associated with a decrease in diarrhea and otitis media and in infants, and with a decrease in prevalence of being overweight or obese in adulthood. Regarding the infant diet effect on the microbiota-gut-brain axis and on the microbiota firstly, the lower fecal alpha diversity have been reported in breastfed infants compared to formula-fed infants. In intestine, human milk had some effect on gut health, but some data still controversial regarding the modulation on gut permeability and immune response. And finally, at brain level, breastfed infants had better language and skills functions compared to formula-fed infants, and that these breastfed infants also had better cognitions compared to infant formula-fed infants. All these different studies were conducted individually to observe the diet effects on one part of the microbiota-gut-brain axis, but no integrative study on the whole microbiota-gut-brain axis have been done. To complete literature data, we had two main research questions. In this study, the first one was, does the infant food modulate the digestion behavior and the dietary nitrogen and amino acid, especially tryptophan, bioavailability? And the second research question was to know, how does the infant food shape the microbiota-gut-brain axis, including tryptophan pathways? To answer to these two research questions, we conducted an animal experiment using 24 Yucatan piglets that firstly received during 8 days an adaptation diet made of a raw cold milk powder enriched with vitamin and minerals. And after, piglets were randomly assigned to one of the specific diets fed. The first group received during 6 days a pasteurized human milk before receiving fresh human milk pool on the last experimental day. A second group, called infant formula group, received during 6 days an infant formula. And finally, a third group received during 3 days a protein-free diet for endogenous loss determination. All the different piglets were then euthanized 30 minutes after the last meal for blood, digestion, and tissue collection. If we are focused now on the diets fed, the human milk group received firstly a pool of 22 pasteurized human milk samples before receiving a fresh human milk pool made of 15 to 20 fresh human milk samples. The second group received a standard infant formula powder rehydrated in similar conditions to what parents can do. And if we are looking at the diet composition, we had a lower crude protein content for fresh human milk and infant formula compared to the pasteurized human milk, with no difference observed between fresh human milk and infant formula, and that the true protein, lipids, and tryptophan content was lower for the fresh human milk pool compared to the infant formula and the pasteurized human milk. A third group was also fed a protein-free diet to determine the endogenous loss, and all the different diets, the pasteurized human milk, fresh human milk, infant formula, and protein-free diets, were all enriched with cobalt EDTA, which was used as an undigestible and unabsorbable marker for digestibility measurements. To determine the digestibility, we use the calculation of the true ileal digestibility presented in previous work, which was made of the ratio of the differences between the dietary amino acid intake and the exogenous amino acid flow regarded to the dietary amino acid intake. And this exogenous amino acid flow was composed of the total amino acid flow in ileum, which was the amino acid contents in ileum related to the amino acid intake using the marker in diet and ileum, to which we subtract the amino acid endogenous flow in ileum, and this was determined using the protein-free diets. It has been shown that this digestibility measurement was a good estimation of the amino acid bioavailability by the FAO. So if we are coming back to the in vivo experiment, we collected some tissue and digesta in brain over 6 areas, but also along the digestive tract from the stomach to the rectum for fecal and microbiota determinations, but also in liver and plasma. All these different tissue and digesta were all characterized through different analyses to determine and qualify the in vivo and in vitro digestion kinetics, but also the microbiota-gut-brain axis by analysis conducted in the Institut Numécan, but also in Redet Institute and Ag Research in New Zealand, where I went for processing the data, but also for some digestibility measurements in the STLO mainly, after the development of the tryptophan determination method. So now I will present you the results of the first main research question, which was, Does the infant food modulate the digestion behavior and the dietary nitrogen and amino acid, especially tryptophan, bioavailability? Firstly, I present to you here the diet structure along in vivo digestion, and we will begin by the diet structure before digestion. So here I present you the confocal microscopy images of the diets before ingestion and the particle size distributions of each diet. And we found that before ingestion, we had a higher particle size in human milk, and it could be explained by the presence of some human milk fat globules in comparison to the infant formula submicronic droplets. In the gastric digester, we also found a higher aggregate size with human milk compared to infant formula, and it could be explained by the different heat treatments applied. Indeed, we had a raw human milk pool compared to heat treated infant formula. To better understand what happened for protein digestion, we choose to compare in vivo and in vitro data obtained after the digestion of the fresh human milk and the infant formula. For the in vivo data, it was obtained 30 minutes after ingestion of the last meal in piglets, and for in vitro data, it was obtained using the in vitro dynamic digestion model DG. So we were interested in the residual gastric intact proteins, and we found that for lactoferrin, beta-lactoglobulin, and alpha-lactalbumin, we were in similar range between the in vivo and in vitro digestion models, with no difference between the diets obtained in vivo or in vitro for alpha-lactalbumin, but that for caseins, we had difference. Indeed, despite of no difference observed between diets during in vitro digestion, we had a significantly higher residual intact casein percentage with human milk than with infant formula in piglets. And we also see here that for infant formula we are in a similar range between human milk, between in vivo and in vitro digestion models, but that for human milk we had a higher residual intact casein percentage during in vivo digestion than in vitro digestion. That difference can be explained by the heterogeneous in vivo gastric emptying of human milk. We found furthermore that we had similar peptidomic profiles between in vivo and in vitro digestion, showing that the present in vitro dynamic digestion model allowed the investigation of protein digestion kinetics. If we are now coming back to the diet structure along the in vivo digestion, we saw that between stomach and jejunum, we had a faster protein deconstruction with infant formula than with human milk, and that in the jejunum of human milk fed piglets, some human milk native fat globules remains. We also found that there was no difference in ileum and colonic digesta between the two groups, and that for colon digesta we found some protein contents that mainly can be explained by the presence of some endogenous protein. Now we will make a little focus at the ileum phase, in which we measure the digestibility. If we are looking at the nitrogen digestibility, we found a lower total nitrogen through ileal digestibility in human milk than in infant formula, with no difference observed for amino acid nitrogen. This different nitrogen digestibility can be explained by the non-protein nitrogen fraction of human milk, which was higher than in infant formula, and that seemed to be less digestible and absorbable in human milk than in infant formula. The non-protein nitrogen fraction of human milk is notably composed of urea and N-acetylglucosamine coming from the human milk oligosaccharides, and it has been shown in literature that these two previous components had some bifidogenic properties. So today, the bioactivity of non-protein nitrogen fraction is poorly considered in infant formula formulations, and these results led us to ask a few more questions, notably regarding the physiological role of the non-protein nitrogen fraction and its effect on the microbiota. Now, regarding the trial digestibility of amino acids, we found that we had a high amino acid trial digestibility for both diets, with similar digestibility obtained for most amino acids, including tryptophan. But we still had some differences in digestibility for some amino acids, such as lysines, which digestibility was higher in human milk than in infant formula, and that for alanine, phenylalanine, proline, serine, threonine, and valine, their digestibility were lower in human milk than in infant formula. For the later amino acids, it could be due to the fact that they are highly represented in some proteins, especially the lactoferrin and lysozyme, that had been shown to be resistant to digestion, especially in the native form. The trial digestibility was used to determine the digestible indispensable amino acid score and evaluate the protein quality of both diets. So as presented in the context presentation, the DIAS is determined using the amino acid content and the amino acid digestibility, and they are both compared to the amino acid reference protein, which is the human milk aminogram. So in the present study, the DIAS of human milk was higher to that of the infant formula, and it was in agreement with what we found in literature. In both cases, for human milk and infant formula, the DIAS was related to the aromatic amino acids, the phenylalanine and tyrosine. Despite of the lower DIAS obtained for the infant formula, the present infant formula had a good protein quality. Now I will present you my results of the second research question I had, which was how does the infant food shape the microbiota-gut-brain axis, including tryptophan pathways? To answer to this second research question, we conducted a multiple factor analysis on 870 variables coming from different analyses presented here and grouped by functions, for example, immune or barrier functions, and by sites from intestine and brain, leading to 26 groups of variables. This statistical analysis conducted to obtain this plot of individuals, showing that we had two distinct groups depending on the infant feeding, and that the two groups are mainly explained by the dimension 1. When looking at the plot of groups, of variable groups, we found that almost all the different variable groups contribute to explain the differences between the human milk and the infant formula group. Firstly, we will focus on the microbiota compositions that can explain a part of the difference observed between the human milk and infant formula groups. We firstly choose to study the alpha diversity using the Shannon index to see the diet effect on the microbiota composition, and we found that during the adaptation period there was no difference in alpha diversity between human milk and infant formula piglets, and so we begin with the same initial profile, but after 5 days of a specific feeding we found a decrease in alpha diversity with human milk in festes and colon, with no difference observed in ileum. This decrease in alpha diversity with human milk was also observed in festes of breastfed infants compared to infant formula fed infants. If we are Now, looking at the beta diversity using the Unifrac index, we also found two distinct groups in colon, but no difference in ileum. Regarding the taxonomy and the phyla level in colon, we found a significantly lower bacteroidetes relative abundance with human milk than with infant formula, and it was in agreement with what was found in literature in breastfed infants. These data highlighted the role of human milk components in the microbiota shaping. Indeed, it has been previously demonstrated in literature that some human milk components, including human milk oligosaccharides, urea, lactoferrin, or milk fat globule membranes, had an effect on the microbiota modulation. Moreover, the different microbiota variable groups, we also found that the immune and barrier variables contribute to explain the difference observed between the human milk and the infant formula groups. If we are looking firstly at the barrier function in intestine and especially in colon, we found that genes encoding tight junction protein were less expressed with human milk than with infant formula, and it was in agreement with the paracellular permeability determined using Ising chambers. Indeed, the paracellular permeability of the human milk group was higher to that of the infant formula group, but it's still in a physiological range. This data highlighted that the human milk modulated the kinetic of maturation of the epithelial barrier and was in agreement with what found in the previous study in some milk-fed piglets. At brain level, genes encoding blood-brain barrier functions in striatum were also less expressed with human milk than with infant formula. When looking at the immune functions in colon, we found that over all the genes encoding immune function studied, 48% of these were significantly differentially expressed between human milk and infant formula feeding, with for anti-inflammatory, B cells, pro-inflammatory encoding genes, or receptors and cellular interactions encoding genes, they were all more expressed with human milk than with infant formula. All the genes presented here were also positively correlated with Enterobacteriaceae and Veillonella, which were more concentrated and more abundant with human milk than infant formula. To briefly conclude on the diet effect on the immune and barrier functions, we found that human milk boosted the immune system, such as previously reported in human milk-fed young pigs, but also that it exists a relation between the increase in permeability and immune system maturation, as previously described. We also found that it exists some correlation between the microbiota and immune response encoding genes, showing the important role of the microbiota, notably regarding the induction of immune system maturation during breastfeeding, but also in preventing immunodependent diseases in adults, as shown by Al-Nabani et al. In their work, but also that we only had a few information for barrier and immune response encoding genes in brain. I will now present you the results of the diet effect on the tryptophan pathways in intestine and brain. Before presenting you the different diet effects on the tryptophan pathways, you have to know that the dietary tryptophan intake tends to be lower for the human milk group than the infant formula group. I represented here the different tryptophan metabolic pathways with in cycle the different genes assayed and in box the metabolites assayed. So in colon we found that the quinuronian pathway seemed to be favored and preferred in intestine with human milk compared to the infant formula feeding, but that the serotonin pathways and the tryptamine productions seem to be favored with infant formula than human milk. We also found during the study that it exists some positive correlation with the pro-inflammatory cytokine that were more represented in the colon of human milk-fed piglets with the IGO, and it was in agreement with what was previously published, but also that it exists some positive correlation with some colonic microbiota with 2 other enzymes involved in the Comunian pathway, and this genera involved in the correlation were also more abundant in the human milk group than the infant formula group. Furthermore, we found that in plasma, despite of the similar true allele digestibility of tryptophan between the 2 groups, we had a lower tryptophan content in the plasma of human milk-fed piglets compared to infant formula-fed piglets. At brain level, we found that human milk enhanced the tryptophan and its metabolic pathway, especially in hippocampus and brainstem. We also found some positive correlation between the serotonin receptor encoding genes in hippocampus with the ileal and colonic microbiota. To briefly conclude on the tryptophan pathways, we found that the infant diets led to different tryptophan metabolism, with at intestinal level the kynurenine pathway favored in human milk group, while the serotonin pathway was favored in the infant formula group, and that at brain level the tryptophan metabolism was enhanced in brain of human milk-fed piglets. To conclude on the present study, we highlighted that we had differences in infant diet composition, but that we had a similar digestibility for amino acid nitrogen and tryptophan. We also highlighted that it exists an undigestible and unabsorbable fraction of the non-protein nitrogen in human milk that is poorly considered yet in the infant formula formulation. We also determined a higher DAS for human milk than for infant formula, but the present infant formula still had a good protein quality. To answer to the second research question, we highlighted that the infant nutrition differently modulated the microbiota-gut-brain axis through the modulation of the microbiota composition, especially due to specific human milk components, but also that the infant nutrition differently modulates the epithelial barrier maturation and the immune system. We also determined that infant nutrition led to different tryptophan metabolic pathways and that it exists some correlation between the gut microbiota and the gut and brain genes and metabolites. So to conclude on this presentation, we highlighted that human milk has an important role on the microbiota-gut-brain axis development. I would like to thank the Infogest Viva Webinar to let me the opportunity to present you today my PhD work, and thank you for your attention. I will be very pleased to answer to your questions.
Okay. Thank you, Elise, for your very nice presentation, and also congratulations for all the work.
Thanks.
It's really, it seems to be a lot of work.
Yeah.
And also apologize for the technical issue. So we have some questions for you. Maybe before to start with the question, we wanted to ask you if you have you ever thought on your future perspectives about maybe if you are looking for maybe a job in industry or maybe for a postdoc?
Yes, I'm looking for a new opportunity in France or abroad for after my actual postdoctoral position, and I would like to stay in a field related to nutrition and specific population regarding the food industry, mainly more than the research in overall.
Okay, thank you very much. Now, so we have two questions from Sofia Strugo. The first one is, how was the residual gastric infant protein determined from the in vitro digestion samples?
Okay, so the results we had for residual intact gastric proteins were obtained using SDA stage analysis, and we corrected the meal dilution with the non-leucine and used as a marker dilution.
Okay, thank you. So we continue with the second one. Given the positive and negative results associated with the human milk intake, do you recommend that feeding a mixture of human milk and infant formula is better?
I think that the most important thing is, despite of positive and negative results associated with human milk feeding is recommended by the World Health Organization, and also in France by national recommendations. So breastfeeding is the first things to do, but if you cannot or it's difficult to breastfeed, it's better than nothing. Like, it's only 1 week, it's better than nothing. So yes, a mixture of human milk and infant formula is better than only infant formula feeding.
Okay. Sophia Strugo, thank you very much.
You're welcome.
Okay. We have one more question. Do you think that the results obtained in vivo can be extrapolated to infants?
Yes, I think so, because we use the mini piglets model that has been already shown to be relevant to what happened in infants, and also because we found some results that were similar to what was obtained in infant study, notably regarding the microbiota, but it still have taking with precautions because it's only animal model and not the reality. So more research has been to be conducted on other points in infants to be sure that everything we show today are what happened in the real life.
Okay, thank you very much. Another question. Why did you use mostly pasteurized human milk rather than fresh human milk to feed human milk fed piglets?
So we firstly fed piglets with the pasteurized human milk because it was for us firstly easier to collect rather than fresh human milk, and it's also representative of what infants can receive at the hospital. So it was easier, and also because we need a large amount of milk to feed all the piglets we had. So that's why we choose to firstly feed the piglets with pasteurized human milk. And we also wanted to feed piglets with fresh human milk on the last experimental day for digestibility measurements to be as close as we can to what infants receive in the real life and to be very close to what's happened in infants.
Okay, perfect. I think that we have more questions, a lot of questions. Okay. Which of the signals do you think is stronger between the 3 possible protein digestion pathways? One of them is digested, then absorbed, then brain axis. And the another one is the non-digested protein, then good microbiota, then absorbed, and finally brain axis.
Okay, I'm not sure to get all the question. Can I so the different way of the different nutrients, but can you please answer, tell me again the question?
Okay. I mean, I understand, like, you are explaining it about first, the protein is digested, and then one part is like,
available for the organism.
Bioaccessible.
Yes.
And then this is immediately absorbed and directly goes to the brain axis. And the other way is that the non-digested protein pass through the colon
Yes.
And then in the colony is fermented by the good microbiota environment. And then after the fermentation, it's absorbed and then goes to the brain axis. This is more or less that I understand it. I don't know if it's clear for you.
Yes, it's almost that. Like at the end, there is in colon there is some part of proteins that are fermented by the microbiota, but for some they can be absorbed or used in other metabolism, not especially in brain after the absorption, but in the whole body. And also that a very low amount is not digested, and so it still goes through the whole digestive tract.
Okay, so maybe we have one more. Where the gastrointestinal condition uses in the DG system, the dynamic,
Yes.
were similar to the physiological condition of infants.
Yes, we parameter the in vitro digestive model with the infant parameters. So we were very close to what we would like to show.
Okay, thank you. I think that we have time for more questions. Could you explain to us what the criteria you use for selecting that specific infant formula?
So it was the formula made in our lab with the eye treatments, like what one we can find in the markets. And we add firstly a part that we use in this infant formula ideal serum, so a whey protein serum only made and used in this product, and not a cheese serum as we can find in other infant formula. So we might have a better quality with our infant formula than what we can find in others that are not made from ideal serum.
Okay, thank you. We have another one. Can you explain in a very general way what are alpha and beta diversity and how it should be interpreted?
So for the alpha diversity, it will show the diversity of different microbiota units in only one samples and populations, and the beta diversity will be used to characterize the diversity of the microbiota, but between two different groups, like showing the intra diversity and the diversity between two groups, two population. So it's two different elements to characterize only an overall sample or two different samples.
I think we still have time for one or two more questions before the start.
Okay, so we have more. Do you think that euthanasia in the final step of the in vivo study could interfere with the results?
I don't think so, because they were not really euthanized. There were we done the euthanasia by electrical stunning, immediately followed by exsanguination. So normally it doesn't affect what we obtain as results after this euthanasia. We didn't inject anything in piglets.
Thank you, Elise. And I think that we have time for, okay, we have one more in the Q&A chat. May Alot-ID, I think. Did you publish your work?
So I actually done two papers. First one already published on the microbiota-gut-brain axis that's available in Frontiers in Nutrition. And the second one was recently accepted in Journal of Nutrition regarding the trial digestibility of amino acid and nitrogen. And I'm still having to write the 2 other papers on the metabolites parts and on the comparison of in vivo and in vitro digestion.
Thank you. We have time for one more, or you can? Yeah.
Yeah, you.
Okay.
One more.
Okay, maybe the last one. I think that is related to one of the first one. Could you set out recommendations with all this information generated in your thesis, possibly to encourage to keeping the human meal feeding as much as possible instead of formula milk?
Yes, I really believe that what the results we show here is that the human milk has specific components that are made for infants and that are like natural things and will contribute to an optimal growth of infants. So any time of breastfeeding, if it's 1 day, 2 days, or 3 months or 6 months, are good, and just like to try to breastfeed their infant if it's possible, and even if it's only for a very short duration, it's still very good for infants.
Okay, thank you. Thank you for all your answers.
Thank you very much, Elise. We will finish the webinar just by presenting the last, so the next speakers. So Ever, if you can share your screen, just presenting the slide of the beginning. So next month, we will invite Marcos Infantes Garcia from KU Leuven in Belgium, and he will discuss about from gastric to small intestinal lipid digestion, the role of oil in water emulsion design to affect in vitro lipolysis kinetics. Then we'll have a full agenda until the month of August. And in August, we have applications still available. So just email us at infogest.viva@gmail.com. I hope you enjoy this webinar. Elise, it's really great job on this. I wish you all the best in your future career and to find, I'm sure, a really nice postdoc position.
Thanks.
In the future. Thank you, everyone. We had 50 participants today and attendees. We have kind of 400 views on each YouTube videos. Don't hesitate to subscribe on the Infogest channel on LinkedIn. And I'm saying to you, see you next month.
Bye.